thank you again to you and your team for giving hope to ALS patients. I would now like to welcome Adrian Ivinson, who will moderate our panel in a discussion on SOD1 silencing therapies. Thanks a lot.
Thank you. Sorry. I'm just going to start off by looking at that camera over there and saying a few words to Avi. Avi, congratulations on your marriage, on your new wife, your daughters, and on this fantastic prize. You brought it all here. You started it. You stick with it. You're an inspiration. So if I had a glass of fine malt whiskey, Avi, I'd be raising it to you right now. Cheers. So what we're going to do, and if I could invite the panel to join me, please jump up. What we're going to do just in the next 15 to 20 minutes is expand the conversation a little bit around ALS treatment strategies, the field in general, where it might go next. And to help me with this, Lucy's first, then Mikhail, then James, then Ed. It doesn't matter.
It's Ed and then James. We adopt any name.
So to help us with the conversation, I've very informally introduced them there, but let me start. You've already met Dr. Brun. Lucy is the Chief Scientist and VP for Research for the ALS Association. Next to her is Dr. Michael Preminger. Michael is an Executive Director and a Director of Business Development with the Harvard Office for Technology Development. Next to her, we got it right, yep. There's Ed. Ed is the CEO of Sarepta Therapeutics. Sorry, Ed, I butchered the name. Could you?
It's Sarepta.
Sarepta, thank you. And is a long-term, big and medium, and now I guess small pharma scientist and a neurologist. And next to him is James Berry. Dr. Berry is also a neurologist, and he's the co-director of the Mass General Hospital ALS Multidisciplinary Clinic, and delighted to have them all with us today. So what we're going to do is I'm going to just put a few questions out there to our panel, and I've asked them to mix it up and argue with each other. And if you hear them say something that's clearly wrong, I want you to jump up, and we'll argue with them as well. So, Lucy, let me start with you. We just heard this wonderful presentation and the prize. It's, let's be clear, that was mice, not people, and that was SOD1 mutant mice. And only, a few percentage of the ALS patient population carry SOD1 mutations. So talk to us a little bit about what that means for human treatment and what that means for patients who do not carry this SOD1 mutation.
So I think my first comment, and all of you in the audience will probably agree, having heard over the last day or two presentations in the SMA field and the gene therapy that actually is in clinical trials. So this is no longer an academic exercise. We know it can be done. So that's very encouraging. I think one key difference, and we chatted a little bit before, is that what is in trial for SMA is the AAV9, and so this is AAV10, but that might not be that difficult to translate. And so yes, it's only 2% of ALS, SOD1. We know how excited we are that there's an antisense trial by Ionis in the 2% ALS for SOD1. And I think that it's really important to have a success for ALS. It doesn't matter if it's a small percentage. We recognize it's the right target, so that the opportunity for success is huge. And having that success, I think we will then have an opportunity to use that same technology for several other targets. So in a way, we set the stage for later discussion. So I think there's a huge place for having a success in a small percent. And I think that we really know that it can be done. I think we should highlight the fact that there are others in the room. Voyager Therapeutics is here with us, using gene therapy technologies in different ways. And so we're excited, and we hope that with these different strategies and slightly different approaches, we will come to the home run of finding a successful therapy. So I applaud the prize, I applaud Avi for his vision, and I think we're so much further to the next step.
Yeah, thanks, Lucy. Michael, I'm going to turn it to you. You work and live every day in that rarefied world, that desert that Nicole referred to, and you work in the sort of business development and that sort of translation field. What do you see in this prize? You work with between academics and pharma and biotech all the time. What is it that the industry might see in this development that you think could be a positive sort of push for them? Why would they get excited by it?
So maybe I'll start by just reminding everyone, that the desert today is much more flourishing than the desert that we actually started working in when we defined this prize, where ALS was really a foreign word and certainly not a disease that anybody was interested in. And maybe I'll start talking a little bit about the academic side of things. So in the lack of invitation for innovation in particular fields by a funding entity or some other motivation, I would say that innovation is driven pretty much by, at least in this country, by NIH agenda. And so when we started, ALS was certainly not on the NIH agenda, which means that innovators in the academic labs were trying to direct their research and write their grants as focused on things that are on the agenda. So if we were thinking about neuroprotective targets or motor neuron targets, what we would see is focus on things that our investigators thought would be relevant for NIH or for industry. So they would look at stroke, they would look at, large markets as perceived by them. And I think that the work that Prize4Life has done, which combines, the prize and the incentive and this, putting the spotlight on ALS, together with the very important work that Melanie and Jessica and Sarah did in actually doing the hand-holding and presenting the access to the tools that are needed, has actually shifted the attention. And of course, along with innovation came also interest, and I think we've been lucky that the blockbuster model just kind of exploded in front of our faces just as we were getting ready to bring innovation to companies. And so now it became a possibility to actually take that innovation and put it in the hands of companies. And so, I think that attracting so many groups to create tangible opportunities is part of that maybe perfect storm that I am personally optimistic will lead us to additional breakthroughs in the field. So again, setting the agenda, creating some initial opportunities, and certainly showing for the first time, as Lucy, you said, that it's possible will definitely open up additional opportunities. So I think this has been a very important kind of advance or opportunity to open up the possibilities on both ends.
Thanks, Michael. Ed, sort of leading on from that a little bit, from your seat for many years now, decades in pharma and biotech, this particular prize-winning initiative and this particular model is a genetic model. How important are the genetics to the industry? And what are the lessons that, from your seat in industry, you take away from this? Again, most of the ALS patients don't have this or similarly defined genetic mutations, but nonetheless, genetics plays a huge part in the field.
So one of the challenges I think that a biotech or a pharmaceutical company has is what is the target? Do you actually understand the disease? So if you look at ALS in general, if you don't understand what's the biology, what's the mechanism of disease, what's the target, it becomes very daunting. If you break it up into smaller targets, so SOD1, now I happened to be at MGH East when Bob Brown discovered the SOD1 gene. And that was a great breakthrough. But everybody said, well, it's only a small percentage of the whole group. But yes, it gives us an insight into the disease. Because before that, we didn't understand what the mechanism of disease was. Doesn't mean that every gene defect is going to act the same, but there has to be some commonality. There has to be something that's happening in the anterior horn cell, so it gives us an insight. And I think one of the things about a researcher, a really good researcher, is not to have the common sense to quit when reason and logic tell you you're beaten. That makes a good researcher. You have to persevere. It's not just brilliance in insight, it's perseverance. And I think one of the things that I've been very pleased with, if you think about it, you know, it's really been a very long time when this gene was identified. But having a potential therapy, and we know that gene therapy, I mean, I started back in the early 90s doing gene therapy. My concern is that I wouldn't finish my fellowship before all the diseases were cured. I now have been humbled, and I realize, boy, does it take a lot of work. We have to make small steps, but I think this is a first step in a very important disease. What we learn from SOD1 will help us in C9orf72. It'll help us in the next genetic cause, and eventually it'll help us conquer all of the reasons and all of the causes for ALS, which is, you know, it's a disease that's waiting to be cured, and I you know, really commend Avi for having the insight, because what we need is money and interest, and, you know, as I look at this, we need young researchers, the next generation, to be really interested in this disease, and I'm heartened to see that our young French researchers are interested in this disease, but that's what we need. We need generations of people to continue to fight. And, you know, you have to be warriors in ALS. You have to be willing to fight the fight, because it is a long fight, but eventually we'll win.
Thanks, Ed. Before I turn to James, Lucy, how does the ALS Association approach that as well? How do you, I mean, you're in charge of, you know, supporting so much research around the world. How do you tackle that question of the genetics of ALS and what it teaches us?
So I think we recognize that genetics has really paved the way for everything we've achieved so far in building model systems. I think it sets the stage nicely for the second half this evening, where we've brought in our keynote speaker, who will very much show you how we've built targets from genes found. We find new genes. And I think it's a big picture. Quite frankly, any geneticist in the room here will say that probably most of ALS, if not all of ALS, has genetic underpinnings. And so some, for the cases as an SOD and C9, there is one major gene, but there probably are many genes involved. And so I think these technologies, and I hope we'll highlight that later in the evening, it's so exciting because we now have antisense approaches, we have gene therapy approaches, and we have a large number of increasing targets that are very key. If we say that SOD1 is only 2%, 1 in 10 will be a carrier of the C9 mutations. TDP43, although there are no gene mutations in them, we know that we see that in all cases of ALS, and maybe there are downstream genes linked to that. So I think it's strongly, although environment plays a role, genetics are our clues in, and industry needs targets, and I think we can provide them. So we're very committed to that area.
Thanks, Lucy.
I'd like to maybe jump in for a second and talk about an example in which Prize4Life has actually created an opportunity for a broader platform that was not genetic-based to be applied in ALS. And this is the lead program with Denali Therapeutics, which is a focus on ALS. It is now in phase 1 clinical trials. And it emerges from work that Professor Junying Yuan did at HMS in my jurisdiction. And it was completely focused on other potential indications, but we made an introduction and made testing in mice possible, and push Junying to try. And eventually, this broad platform that deals with cell death is first applied to ALS. So it's just an anecdotal example of the impact, and I'm not sure that it applies directly to what you were saying, Lucy, but it just, I felt like I needed to share with it, and again, thank the team for making that possible, and seeing this going into the clinic, and maybe again taking a complementary approach to the genetic approach, and boldly trying to apply a broader a mechanism-based therapy to ALS is just extremely exciting.
So when we were thinking of this discussion and the panelists, Dr. Berry's comment to me was, "Why did you give me the tough question?" And indeed I did. So it is a tough question. James, works with and takes care of ALS patients every day. He's also heavily involved in clinical trials and clinical research in ALS, and in many ways is closer to the frontline trench than any of us, apart from the patients. James, what is it? Why are we still looking for a treatment now? Why haven't we got one? What's the impediment? What's anything that any of us could do to push it forward? What does it look like from your perspective?
That was the easy part. The hard part of that question was, and what are the solutions? And but I mean, I think we're coming to the solution. So I mean, I guess I would start by, first of all, I think a lot of what I'm going to say is going to reiterate what we've already heard. But I'd start by saying that understanding the biology of the disease is kind of where we see clinical trials going now. That's sort of the starting point, which may seem obvious, but has not always been the case. And that's possible because we are increasingly understanding the biology of the disease. It's complex. We don't understand entirely. Genetics has taught us a lot. I think genetics, our understanding of the genetics has done a lot. It's taught us about the biology of the disease and given us probably a plethora of new targets, which we're now beginning to go after in trials. It also has become a target and in a way gets us around the biology of the disease. So some of the really exciting things that are going on in SOD1 knockdown strategies are exciting because we don't need to understand every pathway that's set off by the SOD1 toxic gain of function, just that there's a toxic gain of function and we can target that. And so it sort of limits what we need to know before we go after a therapy. And I think that's a huge boon and will play out not just for SOD1, but for C9. And the other thing that I think we haven't talked about is that we're on the cusp of understanding genetic modifiers in a really meaningful way, and that will give us a whole new slew of targets, hopefully, for gene therapy. So I think that's sort of one direction that things are going. And what's the result of that is we've taken an uncommon disease and now turned it into potentially many groups of very rare diseases. And I think that's important because it informs what we do as clinical trialists. So I think one place where we've done a very nice job, led by many of the people in the room, is to create a community of clinical researchers and a network that allows us to do multi-center trials. And we've also worked very hard to kind of improve our clinical trial design. I think as we face this change from an uncommon disease to potentially many very small, quite rare diseases, we may need to update our clinical trial designs to sort of match that and look for how are we researching very, very rare diseases, how are our colleagues doing this, because we may be dealing with just a series of those, and we can do that. That's absolutely doable. We just need to sort of say to ourselves that we're doing it. Another place that I think we're making great progress is incorporating biomarkers. And I think the fact that we haven't had biomarkers has really been a difficult thing for our clinical development programs over the years. It's an easy thing to say if we incorporate biomarkers, then our drug development gets easier. It's a harder thing to actually have those biomarkers, but we're making inroads. So neurofilament heavy, neurofilament light, PBR as a PET ligand, EIM, these things can guide us. The other thing I think that we're doing is developing pharmacodynamic markers along the way as a part of the drug development program. And I think that'll lead us through some of these complex biological questions. So, the tools drive our ability to do the science. And as our tools have improved, we have a whole new sort of world open to us to do this better. And then, the last problem that's an underpinning to all of this is funding. I mean, having interested industry partners, having the attention of the NIH, and having groups like Prize4Life and ALS Association funding this work is absolutely, incredibly important to all of this.
Thank you. I'm just looking at my timekeeper and saying, do we have a few more? So let's open it up. If there are any questions that you'd like to pose to any or all of the panel, please do. And whilst you're thinking of those questions, I'd just like to reiterate one point, and that is to all of you who created and drove Prize4Life from the start. There's one thing that in the SAB meetings we've had over the years, and we would have lots of good arguments about almost anything. But one thing we didn't argue about is it became apparent as time went on that this was having an impact on the community. Just more people were talking about it and thinking about it across academia and biotech and industry, and that was clear to all of us. Now, any questions, please?
Yes. Just to take the contrary view for a moment, what if the biology of the motor neuron death following an SOD mutation is dramatically different, not even related perhaps to the biology of motor neuron death not associated with SOD? And might we be putting too many eggs in one basket by going only in the approach where we learn something like silencing when it won't be relevant?
I think by no means did we say, would we argue that the therapy would be relevant for all ALS, because we understand that there might be some common things, but we know that there's something very unique to SOD1 mutations, actually. We know that TDP43, it's controversial, may not be in those cases. We know that it might be slightly different. I think it's technology, though, that can, and again, I don't want to sort of set the stage too much for Dr. Gittler later, but we'll learn how we can potentially think of other genes that have much broader effect to others. But it's the technology that counts, and therefore successfully doing it and having a successful clinical trial where we know what the readout looks like will set the stage for the next trial where it could be a completely different approach. That was really my argument, to say that even the 2% will benefit us moving it into trials and seeing what does success really look like. What is that change in ALS-FRS score that makes us excited? So that's where I was coming from. I think the disease could have many similarities. We know even within one SOD mutation, it's variable. But I think it sets a stage of technologies like these that we could use if we're looking at this particular example.
Yeah, and I think just to follow on that, I think one of the things is that we can't assume that we're going to have a cure at the first therapy. I think what we're going to learn is that it'll probably be multiple therapies that are necessary, different approaches, different mechanisms. And I think it's that knowledge that we accumulate over years. And thinking back, and I remember one, as a pediatric resident, the childhood leukemia, acute lymphoblastic leukemia, was a 95% fatality. Now it's a 95% cure. That took decades of work, very slow, painful work, trying to understand the disease and unravel it. And I think it's the same is true as ALS. We can't assume that this is going to be a home run the first time. And I think it's really a matter of trying to understand the disease more. And every genetic mutation we look at, we look for commonality. We look for, there's things that we learn. It may not, basically translate to all the causes, but it's a slow, painful process that has to be done, and this is a great start.
We wouldn't mind.
We wouldn't mind.
We wouldn't mind, yeah. We have time for one more, yeah.
What are some of the hurdles in developing antisense technology for C9orf72 or using viral vectors to alter C9orf72 expression?
So I think we have so many experts in the audience
Right.
here that I think I wouldn't do it a service, and I think that's a whole discussion on its own. I think there are challenges. I'm looking for my Biogen colleagues, and
Sitting
oh,
next
right
to
there. I think the two of you should have a conversation. clearly there are challenges. I don't know if we want to tackle it on the panel or if we, in interest
That's
of
a tough
time,
one.
have the
Well, I
time.
mean, just in a very general sense, I guess, you know, without getting specific to C9, it's the sort of the idea is sort of following up on Jeff's question as well, which is, are there, you know, is it going to be a one-size-fits-all if we get a gene therapy right and we can treat SOD1 ALS? Or is it going to be exactly the same blueprint to go to other gene mutations? And C9 is a good example. TDP43 is another good example of saying no, it's probably not going to be exactly the same. But the tools that we develop, the things that we learn along the way are going to be.
And I think I have one of the biggest hurdles is coming in on time, right? I mean, if we come very late in treatment with any of these approaches, we can stop it at that point, but a lot of damage has been done. And I think that's so true of any of the diseases, but even for SOD and C9. Yep,
Right.
exactly.
Well, talking about coming in on time, I'm being told that we are just about to come in on time, so I'm glad you two are sitting next to each other and can sort out the ASOs. Please join me in thanking the panel. Thanks very much.
Well, thank you very much, Adrian, and to our panelists for this great discussion. Let's give them another round of applause. So I think we will take a 20-minute break now, and then Lucy will introduce the ALS Association part, the continuation of this program. Thank you.